speaker, Namrata, and I will just briefly introduce her. She is going to talk today on the title of crack propagation in soft materials, and she is a professor at the Department of Mechanical Engineering, Indian Institute of Science, Bangalore. Her research interests are broadly in nonlinear tissue mechanics, cell mechanobiology, and failures in biological materials and hydrogels. Prior to joining the institute, she worked as a postdoc at the University of California, San Francisco, where she collaborated with the cardiac surgeons. She earned her MS and PhD from the University of California, Berkeley, and she has been awarded the Ramanujan Fellowship and Constance Tipper Silver Medal from the ICF, International Congress on Fracture. She's been on the editorial board of Biomechanics and Modeling of Mechanobiology, Cell Adhesion, and Migration journals. She also serves as the founding chair of the Office of the Research Grants at Indian Institute of Science. With this brief introduction, now I will request Professor Namrata to go ahead and deliver her lecture. We will have the question and answers at the end of this presentation, and anybody who would like to ask the questions can type it in the chat box or in the question and answers box. We will take it up sequentially at the end of the presentation. Thank you.
Thank you, Raghu. Thank you, Vikram. And thanks to all of the InSIS organizers for giving me a chance to present. I'm really pleased to see there's such a vibrant community of people who are excited and interested in structural mechanics topics. So I'll begin talking about some new work which has come out from the lab very recently. I'll begin by telling you a little bit about what the lab does. So a lot of, I hope you can see me and you can hear me. So if there is a problem, please let me know.
Yeah,
Thank you.
Everything is fine.
Great. So a lot of my interest in soft tissues began as a student when I was interested in this structure, which is the aorta. It's the largest structure which carries blood from the left ventricle to the rest of the body. What really interested us was what happens to this tissue as in some cases it seems to grow. It seems to grow very large and it sometimes bursts. This is called an aneurysm and it is usually fatal. The tissue is extremely soft, but can undergo very large deformations of about 60 to 80%. And it is not well understood how failures such as this occur in these kind of tissues. We started looking first from the perspective of stretching and the nonlinear mechanics of these tissues, which have various kinds of proteins that are organized in very nice hierarchical architecture inside the vessel wall. We also looked at the flow physics of how does flow go through these very curved pipes and what causes and what are the cues that cause aneurysm to develop typically downstream of this curvature region. In time, we got interested in mechanobiology, which is to try and understand how cells respond to these stimuli. So a lot of the lab looks at these kind of questions. But centrally, what we are interested in doing is to look at how tissue which looks normal can become highly fibrous, which is a very complex chemomechanical feedback that occurs between the cells and the tissue that causes this fabric to dramatically change and can undergo these kind of failures. So today what I'm going to talk about is some of these processes of what we are interested in looking at is how can we change tissues from normal to fibrosis and how does this influence the mechanical properties of soft materials. So we have been trying to understand this for a while. We look at fibers which are embedded in matrices and we stretch it in this kind of a stretcher. This is built by BIS, who have been very kind to us, and we work closely with them to develop some of the instruments that we use in the lab. So typically this includes a tissue or a material which is put in trampoline fashion, and we track a bunch of particles on this through video extensometry, and that can tell us what the stress strain properties are for this material. We've also more recently developed our own stretcher, which sits on a microscope stage, and we can watch collections of cells, which are shown here in various colors, as they are deformed under various kind of stimuli. In turn, what you see on your right-hand side in this picture is the boundary of a cell, and these are the kind of traction stresses that cells exert. And the substrate on which it is sitting is a soft hydrogel. And this is the project that I'll be talking to you about in a little bit. So to tell you a little more on why we chose the kind of materials we did, early work has showed that the properties of artery-like tissues, including skin, lungs, and so on, are very nonlinear. So they typically have these J-shaped curves. And what is very interesting about these curves is the initial region of this curve is dominated by rubber-like proteins. And the end part where you have strain stiffening response is dominated by collagen. And we were interested in both these aspects of how these things contribute to the mechanics of hydrogels. So we initially started by making both elastin and collagen hydrogels, but soon we realized that the problem was a lot more complex, and we decided to focus on gelatin purely. So why do we really need to study hydrogels? So hydrogels are two-phase materials. They include proteins and water typically, and these are excellent for as scaffolds in tissue engineering. What we also know, our cells are exquisitely sensitive to this substrate on which they're sitting. We don't quite understand how cells know that it is sitting on something as soft as 1 kPa or if it's sitting on something as 40 kPa, and how these signals are going from the cell substrate level all the way back to the nucleus. More importantly, we feel that an understanding of the mechanical properties of these hydrogels and trying to control these mechanical properties will allow us to replicate the kind of in vivo environments that are experienced by cells. And the main problem with working with hydrogels is while they can undergo very large deformations, a problem is that they fail almost like brittle materials, which makes it very exciting. So gelatin is a denatured form of collagen, and gelatin forms three-dimensional cross-link networks. They undergo very large deformations. And again, like I said, they fail like brittle materials, like all soft brittle materials like jelly. So gelatin is widely used for wound healing, tissue engineering, even for contact lenses. Drug delivery is a very big place where it is being used, and also to coat some of the implants. Some of the main problems with using gelatin are that one is it's highly variable because we can't control the length of the polymeric chains that we obtain from the powder gelatin that we acquire. So we can introduce chemical crosslinks and stabilize the gel, but we also can't use too much of the chemical crosslinks, which is typically glutaraldehyde, because it's a cytotoxic material. So, but what we understand is these crosslinks in gelatin, in collagen networks, also increase dramatically as we age. So we want to be able to control these crosslinks, and we want to be able to understand how cells respond to these very controlled hydrogel properties. So what we discovered was that methyl glyoxal is a very nice uh, end product which is involved in collagen cross-linking. So we thought, why couldn't we use both this MgO and glutaraldehyde and try and cross-link the gelatin hydrogels? So this work was done by Anshul. And what you see here are two little blocks of this gelatin. One is control, which is gelatin treated with glutaraldehyde alone. And the MgO has been additionally treated with glutaraldehyde. So we know that one of the big questions in soft matter failures is how can we control the two, so how do we increase the toughness of these hydrogels? Because what we know is that materials like our cartilage is extremely soft, like a hydrogel, but it also undergoes very large deformations, but its toughness is almost in the order of 1,000 joules per meter square. Whereas most of the hydrogels that we are able to synthesize in the labs are typically in the domain of about 50 to 100 joules per meter square. So there's an order of magnitude difference in the kind of properties that we have been able to achieve with biological materials. So the field has been trying to increase the toughness of these gels because that's the only way we can use it for load-bearing applications. One idea which has been in vogue for almost about 15 years now is the use of double network hydrogels, where one network acts as a sacrificial layer and the other one allows these very large deformations to happen. However, in this case of our MgO hydrogels, we have only introduced additional chemical crosslinks. So what we anticipate are that you should have a much higher modulus. But the quantification of failures in such soft materials is a little bit challenging from an experimental perspective. So when we look at SEM micrographs, what we see is these MgO-treated hydrogels, they have much larger pore sizes. And we think that is because the gel has been better cross-linked, and so it's able to withstand a lot more of this loading. What Anshul next did was he went and measured the nonlinear properties of these gels using our a Bose electrophorese instrument in uniaxial loading. And again, we see these classic J-shaped curves. The ones which are MgO treated are much stiffer, as is shown, all the curves are on the left. And here you also see this problem that I have talked about is that of variability in the gel properties. So we don't get them all to fail, which is shown by a very quick drop in stress when it fails almost catastrophically. But the amount of deformations that you see in these gels are typically between about 60 to 70% before they fail. We use this initial part of the curve to calculate the moduli, and we have almost 2 times the modulus for MgO as compared to controls. What we next did was we tried to use some of the strain energy functions for nonlinear mechanics. Typically, we use a neo-Hookean model. We also use a Mooney-Rivlin model, which is called MR here, and a first-order Ogden model. And the first-order Ogden model seems to have a best fit as compared to the neo-Hookean, which is shown in this dotted line, which actually doesn't do very well for highly cross-linked gels. The Mooney-Rivlin is also not a good model because if we explore this idea in the context of what was proposed by early on by exploring these Baker-Erickson inequalities, we see that these curves sort of saturate like this. And so these constants C1 and C2, which both have to be positive, won't quite be positive in these cases. So that says that the Mooney-Rivlin models are also not very good for our hydrogels. So this leaves us only working with an Ogden model, which seems to give us the best fit. So what we next did, and you can see these fits over here, we also have explored if the fits that we get and how good are they with the calculated moduli. And sure enough, we see that the first order Ogden model fits best as far as the RMSE errors are concerned, as well as the measured elastic moduli for the hydrogels. So this is our choice of model for gelatin hydrogels. With this, we next wanted to measure failure in the hydrogels. And what we found in literature was this very nice technique called cavitation rheology. So the idea is, I think I should show first this slide. So the idea is simple. So this is the orange shaped blob is our gel. If we introduce a needle, and you can introduce needles of various bores, and these are of blunted tips. So we are only looking at what happens ahead of this needle. So this is thought of as a crack tip of various sizes that you can start in the material. And we inflate this gel with the bubble. We can watch what the pressure is inside the bubble, and we can inflate it at various rates. And as the bubble inflates, it assumes a specific shape and it suddenly pops. So much of this setup was built by a former student, Achu, who is now in Texas I think he's now in some kind of an engineer in Texas. So the pressurization of such bubbles in this elastic gel will cause a rapid expansion of this defect, which is the cavity that is growing inside. And this is an elastic instability, but it is reversible for a small extent of pressure inflation. However, if you continue to increase this pressure inside, then it undergoes ruptures, which are completely unstable fractures and are irreversible. This technique was first seen by Alan Gent, who saw this when rubber sheets were stretched under uniaxial tension, and they would create these holes inside rubber materials, which he called, which was also understood to have similar dependence. But the technique has been also used by others like Dijon in 1979, which tried to see how the amount of polymer and the defect size, how it can be related to the elastic modulus of this material. Several others have explored this idea to come up with scaling models of increasing the amount of polymer and to see how you can relate it to the resulting modulus for this material. What we decided to do instead was we tried to use these needles inside both our gels. One on the left will be the control gel, control gelatin gel, and the one on the right is the MgO gel. We start with various kinds of needle sizes. These are 75 microns, 150, 230, and 320. These are mostly what are available in the market. And we record the pressure inside the gel. And what you see a plot over here is the pressure as a function of time. So what you see is as I load this gel with pressure, and RA is actually the one which is the smallest bore. So the floor size is really small, and so it makes sense that it takes a much higher um, pressure before it pops. And you'll see the pop as a complete drop in stress, which happens at this point in this curve. You see a similar response in all the other gels, but again, there is variability. And like I explained earlier, the variability occurs because we are using biological materials which have variable chain lengths that we could not control for. But some nice things happen when we start doing the experiments with MgO, where we increase the amount of cross-linking. So the gels are tougher. Before I say tougher, they have a higher modulus, and they have more or less initially for these very large needle sizes, they have a linear pressure with time relationships. But as you go for smaller bore needles, you'll see that the curves become highly nonlinear, which is similar to the kind of curves we saw for the pressure inflation results. When we calculate the critical pressure, which is determined as the point just before the pop happens from our experimental system, we can see a very nice relationship. And so what we're showing is that addition of this MgO seems to dramatically increase the critical pressure that is required for popping the bubbles. And again, you have a much higher pressure which is needed for the smaller bore diameter, for the smaller bore needles. MgO samples take much longer for the initiation of the bubble, but when the bubble actually collapses, they occur much faster after they initiate as compared to those in the gelatin hydrogels without the presence of the MgO. So the next point was that how can we come up with expressions for computing the critical energy for failure? So what we have done is we have used the expression for pressure as a change in the strain energy with respect to lambda theta for a spherical bubble which is growing inside this hydrogel. And we have used this W strain energy function for a neo-Hookean material. And you can see that the relation works out to be this very classic relationship where pressure is E into 5/6. And you have these other terms which come in the denominator of 1 by lambda theta and lambda theta to the power 4. So essentially what we have done is we've modeled the growth of this bubble, which is first term is dUE, which is the elastic energy, and the second is the surface energy term, which is, as we see, very important. And I'll try and demonstrate a little later that in fact it seems to dominate much of the failures in soft materials. So we can use this expansion of PDV, which is sigma dV plus gamma dA, which is this expression which has been expanded in this relationship, and then we can write the critical pressure with divided by E, which is elastic modulus, which has a linear relationship with which is shown in this graph. So what we're saying is the maximum pressure for this elastic gel, or when the large syringe is used, is something that we have been able to see. And so, but as we decrease the size of this needle, or we change the modulus, the surface energy term starts becoming much larger. So when we went back and saw that the neo-Hookean was not the greatest of models, but the same relationship you can see is that this GC term tends to infinity as lambda goes to infinity, which is actually the condition for cavitation. So lambda is the stretch, which is the ratio of changes in R with respect to the original R. But when we model this expression, when we rewrite this expression for W in terms of the first order Ogden model, we start getting very complex terms. And these constants have been obtained based on the experiments that we did on gelatin, which was controlled, and the gelatin, which was treated with MgO. So you'll see very quickly that we get, instead of 1 by lambda theta term alone, we get an ln lambda theta. And here we get an lambda theta to the power 2 for the MgO gels. And these are actual expressions of the strain energy density function that we have used in these relationships. So to look at failure under cavitation, if we explore this relationship in the limit of lambda theta tending to infinity for this Ogden material, what we see very quickly is both the PC values and the GC values become unbound. Which tells us that these models cannot really be used to compute the fracture toughness of hydrogels. And it presents a very serious limitation in cavitation rheology. In fact, most strain stiffening material models that are typically used to show the properties of tissues cannot be used in cavitation rheology. So this is like a big issue. This is something that we recently showed in a paper which got published earlier this year. But when we use the linearized expressions and we look at how PC by square root of E now changes as 1 over square root of R, we see fairly nice linear curves. But again, please note that there's a lot of variance here, which is very typical of biological gels, where we have variability in the kind of material, starting material that we have. But in general, what we can say is that the modulus of the MgO hydrogels, which were double that of controls, they seem to have higher values of GC as compared to that of the control hydrogels. And this is what the table actually looks like, that addition of MgO has been somewhat beneficial because we have increased the toughness from about 13 to about 49 joules per meter square. So we seem to have done fairly well. But the problem remains, which is that of brittle ruptures in the hydrogel. And in fact, what we are saying is that the MgO seems to fail much faster after the initiation of the bubble. What you're going to next watch is let's see if this video is, okay, I'm not sure if the video is not playing. Okay, sorry, the video is not playing. I thought I checked it. But so what I'm going to do is show you instead the panel of images. So what you see on the top panel is the inflation taken from about I think it was 2000 frames per second with a high-speed camera. And this is the top row is control hydrogel, and the bottom one is MgO treated hydrogel. And both dimensions are approximately the same of the hydrogels, but we see some very interesting behavior. So the dark line is the insertion of the needle, and what you watch ahead of this is the bubble formation. So you'll see that in the softer gel, we have a much rougher surface that seems to have been created, which grows. And you can see it's only a single projection image, so we're not able to tell, but it's more or less spherical expansion. And this is the maximum collapse of this cavity that you see inside. In contrast, the MgO gel, this is the needle here, it seems to grow almost elliptically. So this is more like a penny-shaped crack, whereas this seems to grow like a sphere, or a spheroid inside the hydrogel. So this part really intrigued us, and also the times that you see over here are telling you that the bubble collapse happens much quicker once the inflation starts inside the hydrogel, and that is when we start tracking it from time t equals 0. So we are saying that what we are showing is a symmetry breaking transition which happens in the same hydrogel with a single change, which is a change in the modulus that we have introduced in this hydrogel. So that really intrigued us on what causes these kind of symmetry breaking transitions. We also mapped how the bubble grows as a function of time. And both, so the one in square is our control hydrogel and the triangle is the NGO gel. They seem to start at similar ways as far as the growth curve is concerned. But what typically happens in the NGO hydrogel is it starts as an elliptical crack, which is the aspect ratio measured as the ratio of major to minor axis of that sphere or the ellipse that we have. Closer to 1 shows that it is more or less a spherical shape. So this starts as an ellipse. It tries to become a sphere, but then it goes through this instability and pops almost immediately. So what we know is, so what we showed was that that there is a symmetry breaking transition that happens. Cavitation rheology is useful, but it cannot be used when we use complex strain energy density functions that include strain stiffening, where we cannot model these cavitation behaviors. We also think that both the elastic and the viscous properties of hydrogel are critical processes. And so we wanted to next explore what happens. Uh, Another challenge with using cavitation rheology is we can very easily quantify a lot of these behaviors, including Anshul has been doing experiments in cornea, which is a really nice hydrogel-like material, and where he's able to generate these bubbles and quantify how tough corneal tissues are. But again, the problem is that we are not able to see the processes that happen ahead of the crack tip, which are very critical for us to see how to increase the toughness. So that presented a big challenge, and we decided to next try and understand what happens in the vicinity of the crack tip. So since cavitation rheology wasn't going to help us, we went back and tried to look at pure shear samples. So we have created sheets of both these hydrogels. One would be the gelatin, and one would be that treated with the NGO. You'll notice that the dimension of the sample is much longer along the clamped edge as compared to the edge along which we have introduced a notch that you see over here. And this is the actual picture of what the gel looks like. And this sample has been spray painted because I will show you data from the strain fields that occur ahead of the crack tip a little bit later. So this kind of an experiment allows us to control this initial crack size, which we do with a very sharp blade. We can also test various strain rates, and we can look what is happening ahead of the crack tip if we have a camera mounted above this sample. So what we next did is we tried to use DIC with NCore, which is an open source digital image correlation method to quantify the strain fields and hence the failures in these kind of hydrogels. Um, for this material, the strain energy density is calculated experimentally by first subjecting an unnotched sample. So we don't have this notch here, and then we repeat this experiment in pure shear constraint, and we can calculate what the critical energy release rate is by this expression given in this graph. So one of the critical things that we realized is we needed to better understand what is what is happening at the tip. And so you see these two samples where the crack has run along, and this is an actual digitization from one of the images of the crack inside the gelatin gels. And you'll notice that ahead of the crack tip, the curve is highly distorted. So a common method to calculate curvatures is by using a parabolic method, which is shown in this dotted line. But the point is that the parabolic method might work in some cases, like here, it's not so bad. But if we are looking at what is happening at the tip, this parabolic method is really very poor for these kind of sharp cracks which happen ahead of the crack tip. So we needed to come up with a better method. So Anshul has worked with Supreet on this work. And what we did was we looked at single point xi where we want to calculate the curvature. We look at a bunch of points before xi and after xi and use a Taylor series expansion and can write an expression for the kappa, which is the curvature, based on this kind of a function over here. Okay, where I can be any variable, it's essentially looking at some kind of a spline fit. Typically, we use about 5 to 7 points to determine the curvature, and that seems to give us a really nice expression, which for the curvature, which is significantly different from that of the parabolic method. So once we have looked at, so this is the errors that we have estimated, that the parabolic method gave almost 40 to 75% error as compared to the error calculated using our curve fitting method. With this, we went and did the experiments for stress strain, and what you see in the dark line is the unknot sample for the control hydrogels, and this one on the right hand side is the one for, let me get back my laser pointer. So the one on the right-hand side is the are the data for the MGO samples. And again, the dark line corresponds to the unknot sample, which makes sense because here we see that the unknot sample has a much sharper stress versus stretch graph, whereas that of for control is showing much less. The second thing is there's about 4 different samples in the control and the MGO gels, which were notched and tested. And we see some very nice behaviors here. And I want to walk you through some of these ideas here. So initially, the crack seems to grow, and there is a very sharp drop in the stress. And it's very sharp, and again, it grows. And there are a lot of these steps that we see in both the curves consistently in all the samples before the sample completely, the crack completely runs through the sample and tears the gelatin sheets. Before that happens, we also see many little steps that additional steps that we see with MGO crosslinks as compared to the control, compared to the control. So one is that the control samples are failing under lower stresses. Second is there's many undulations of this crack as it goes through the sample. And third is that the MGO samples have many more undulations as compared to control. So these are some things that I'll revisit in a little bit, but we used our expressions for GC and compared it with the cavitation data. And in fact, they look kind of similar for MGO, but for controls, we have almost doubled the value of what we have estimated using this method. There has been no standardization of the various techniques which are out there to measure GC for soft materials. So this serves as one kind of a calibration using the same student and the same set of hands and the same kind of materials. So we are showing that there are some other issues that we see with cavitation rheology. But moving on, Anshul and Supreet went ahead and quantified the failure processes ahead of the crack tip using the dotted sample with DIC. And what you're seeing here are the strains in the Y direction. These are the shear strains, and these are in the XX direction. So typically the Y direction, so this is as we are pulling the sample, that would be Y, and this is X in this direction. So this panel is for control samples, and this one is for MGO. So first we'll start with the shear strains. So what we see ahead of the crack tip are regions where you have almost a pure shear sample. You have some spurious values which come in certain regions, and especially in the edge effects of both this and the MGO hydrogel. Typically, and these are points taken when we saw those step loads progress inside the sample. And you see that the sample quickly tore and went up to this point. And again, we see a very similar pattern of shear, which is maintained inside this hydrogel. And a similar one, slightly lower value though, which happens in the MGO hydrogels. YY strains are uniformly spread throughout the sample, and there's hardly any XX strain that we can detect. That's much smaller as compared to the YY and the EXY direction. These were performed using NCore, which is an open source MATLAB-based software to compute strains ahead of the crack tip. And the shear strains are nicely localized ahead of the crack tip. And again, they occur at about plus and minus 45 degrees during crack propagation. Failures are occurring much faster in MGO gels, which can also withstand higher loads. And the failures through the sample width were very nicely controlled through the MGO samples as compared to these control hydrogels, which had only glutaride treatment. So here's a video which shows how the normal strains EYYs are changing in both the control and MGO hydrogels. And what you start seeing is that there are various zones of strains that develop ahead of the crack tip as the crack tears inside the sample. And we see these sharp variations, and we also, so this was our sample, and so you can see along this line where we plotted the Eulerian strains, we see that just before the crack tip is this zone A, where there is not much happening. So you have this almost zero region here. In this domain of B, we have a very complex evolution of strains that occur exactly ahead of this crack tip and reach a maximum, and they start plateauing out. This is the EYY, and these are the EXYs in blue, which are the shear strains. So based on these variations, we have delineated these domains of ABCD inside the sample, and we see a similar zone which also occurs for the NGO samples. So what this tells us is, based on this plot, that the very nice control that we got during the tear of the sample is dictated primarily by the pure shear state of strain, which occurs in the YY direction, and actually the EZZ direction, which we can't see, but it's orthogonal to the direction of strain. And this is how we have created this pure shear inside the sample, which works very nicely for soft materials. The second thing we noticed was that we started watching what you see on the first plot is the crack length as a function of time for control samples. The one below is for MGO. And we see that the crack length remains more or less constant inside the material. So it takes a really long time when the load hits a critical value, and then your crack runs away inside. And there's again a hold time. And again it runs, again a small hold, and it continues until it tears throughout the sample. And we see this pattern preserved in the MGO gels. What we next did was we quantified the crack velocity in this for this sample. And again, we see that at these points, which are labeled A, B, C, D, the crack velocities are extremely high, which is what we have denoted as a box in this picture. The final plot refers to the curvatures which we calculated using our curve-based spline method. And here's something that we see very interesting as far as the curvature is concerned. The same 4 points ABCD are marked here as in this point, and hopefully you can see them. And the one below, which is for the number of different points where the sample in MGO tore. And what we see consistently is that the curvatures are the lowest at the points when the crack runs away inside the sample. So one, that the cracks propagate after a very long stall, we get the cracks to propagate. And at those crack propagation speeds, the speed is extremely high at those points. And last, that the curvature is the lowest at each of these different points, which shows us that unlike metals, where the crack propagates when it is at its sharpest points, here we have something very counterintuitive, that the crack seems to have that most of the energy that we are supplying, the mechanical energy due to stretching, seems to have been used in absorbing and blunting this crack tip. And when the crack is at its most blunt point is when it starts propagating once again. So this didn't seem to, so we discussed quite a lot about this, and we chatted with many of our colleagues, but this wasn't something that was well shown in the literature. What we also showed was that ahead of the crack tip, and on the left that you see are the control samples, and on the right you have the MGO samples. And again, these are the splotches that we have marked for the DIC experiments. And we see that in addition to the crack tip blunting that I mentioned earlier, we also see some crack bridging mechanisms and crazes, which you see over here, that are very characteristic of polymeric materials, and in fact, a source of toughness in polymers, where we have created these zones of what happens ahead of the crack tip as a mechanism of toughening of this polymeric material. Anshul has done additional set of experiments, which is using a single edge notch. So he's reversed the direction of how these samples were loaded. And I won't go into details, but interestingly, these are again showing a GC values for both the control and MGO gels, which have very similar values to what we have seen earlier. But again, the expressions are slightly different because of the dimensions that we have maintained in the sample. Also, this doesn't work as well because we can't get a very nice controlled tear. So actually, visualization of the crack tip processes are a little more tricky in the single edge notch samples. So to summarize the first few parts of the study, that we've looked at three different methods: cavitation, rheology, the pure shear notch test. Both these methods I've discussed in a little more detail. I broadly glossed over the results from a single edge notch test. So to contrast these three different experiments, in this one, a needle is inserted to create a flaw. It's difficult. Also, I didn't mention how tricky it is. Not all students will have success because a lot of the time the bubble seems to propagate along the needle gel interface, which is the weakest. And so it's always difficult to have a leak-proof insertion until you come up with clever techniques and you know what your gel looks like. So Anshul seems to have perfected this method, which is why we're getting very fast data with this technique. It is extremely useful when the sample when, for example, sample dimensions are limited, like I mentioned in case of cornea, where he's trying these experiments. But it is also limited in the sense that we can't use this method for any material which has strain stiffening response. And so the results can sometimes be quite different from what you can see with a pure shear and single edge notch. I myself don't like the single edge notch tests in soft materials because one is it's difficult to clamp, and because this is a very long edge, then the sample sometimes fails under its own bulk, under its own weight. In contrast, these experiments of pure shear are much cleaner to do, and they're more controlled in how the flaw or how the crack propagates inside this material. We have eyes ahead of the crack tip, and we are showing some very novel mechanisms. These are comparatively easy to perform. They require much longer dimensions and a better ratio of this length is to width. And we are showing that unlike metals, soft tissues have additional terms that can contribute to failures. So when we finally compare the GC for MgO gels, they're sort of in the same ballpark. But in control gels, we see that these pure shear notch and single edge notch gives us similar readings, whereas they're completely different for cavitation rheology. And we're not sure of the reasons why it could be. One thought is that the whole models that we are used is based on a linear model, and that doesn't really work so well as far as cavitation processes are concerned. So these are some of the novel features that we have said, that the blunting of the crack tip seems to be a very nice feature in soft materials, and the cracks are propagating when the tip is at its most blunt. And cavitation rheology has this dominant contribution of the surface energy term. So we looked more at the surface energy term, and here's a nice quote from Wolfgang Pauli, which I didn't know about before we started this work. He says, "God made the bulk, surfaces were invented by the devil." And so what we are actually looking at are what are the contributions to failure from the surface energy term, which seems to overwhelm most of our understanding of how failures happen in soft materials. So I will now tell you a little bit about where we have taken this study. And now Anshul has teamed up with Imnutoshi Jamir, and Imnutoshi is an MTech student. And we essentially sought to answer the question of how can we control gels to have a similar modulus so that we get more or less the same contributions to the kind of how we can feel the softness. And so the linear terms are sort of controlled, but we can have differential contributions of viscosities. And this is a very intriguing way of looking at gels and failures, but it allows us to control both the strain rates as well as the inflation rates that we can control inside this material, and look at the processes ahead of the crack tip. It took us a few iterations, and this time we didn't want to work with gelatin because of the standard issues of not having proper control on the various parameters to give us more standardized results. So we decided to go back and look at polyacrylamide, and these are nice synthetic polymeric materials. They can be made as soft and stiff, and a lot of our other work in mechanobiology, we use polyacrylamide very routinely to make 10 and 20 and 30 kPa hydrogels that we study. So in this case, what Imnatoshi and Anshul did was they used acrylamide and bisacrylamide mixed in water, APS and TEMED as before, that were catalysts to initiate the polymerization. But this time we decided to add more long linear chains of polyacrylamide inside the hydrogel, inside the elastic gel, and then we can increase the viscous component of the hydrogel. So we use the same molds. The dimensions of the sample are more or less the same as what you've seen earlier. When we did the experiments for compressive moduli, you can see that we have, these are all about, I think, 4 samples that they've tested, and there's hardly any variance in the properties of the gel. So you can see there's almost nothing, and all of them are getting very nice reproducible results for what I will call E gels, which are elastic gels, and the VE is where you have more linear polymers, which is inserted inside. And so the moduli are more or less similar. There's hardly any variance. There's a little more variance in the VE gels because of how we've added the viscous component of the hydrogel. One another feature is that the elastic gels are very soft and they're almost transparent, whereas the viscoelastic gels have a little more milky look, and you won't be able to easily see it, but I'll show you some high-speed videos where that starts becoming more obvious. So what we next did was we wanted to look at what were the differences in the storage moduli and loss moduli of these materials. So we used the DMA instrument in our lab, and we measured these properties over a range of almost 0.05 all the way up to 70. Beyond 70, we see a resonance, and so we're not able to use the properties, and so we terminate our experiment at this point. This graph shows the storage moduli for the elastic gels, which are slightly lower than that of the viscoelastic gels, which you see a little higher. The loss moduli, so we put a forcing function of displacement or strain as epsilon naught sine of omega t, and we watch how the stress changes as a function of this forcing function, forcing frequency. So if we have delta close to 0, we have a gel which is more or less elastic. If delta is 90, then we have almost a complete fluid-like response, which we don't have because we can hold and work with these gels. So delta is a small value, which is something that we need to estimate with this experiment. And so we can calculate the tan delta, which tells us the ratio of the loss to the storage moduli of these gels. And you can see how it varies as a function of frequency. We see that from the DMA experiments, the viscoelastic gels had almost 10% higher than the elastic gel at very low frequencies, but this difference increased to almost 20%, 21% at 70 hertz. Whereas the losses were significantly higher at 37% at 0.07, and this difference went up to almost 50% at 70 hertz. So we next did our favorite cavitation experiment, and here we see again, this time luckily the video is playing, thank God. So here you see that the bubbles, initially if you remember, the bubbles were elliptic in the NGO hydrogels, whereas here in the more elastic gels, they grow almost elliptically or a penny-shaped crack. Whereas below you see the bubble inflation inside the viscoelastic gel, and these are inflated at about 200 ml per minute. And you can see these very rough surfaces that evolve in the viscoelastic material. But again, we see this very classic symmetry breaking transitions, which go from an elastic to a viscous from a penny shaped to a spherical, or vice versa, which is the way we think of how symmetry breaking transitions occur. And so we're very intrigued about these transitions. So to explore this further, we started doing these experiments to watch the shapes in these elastic and viscoelastic gels at various inflation rates. So what you see from left to right are inflation rates controlled from 500, sorry, it was microliter, not milliliter. So it was 500 microliter all the way up to about 8,000 microliter per minute. And this top panel is for elastic, whereas the bottom panel is for viscoelastic. So regardless of the inflation, so we see some, again, very interesting findings which haven't been shown earlier, that the viscoelastic gels have very rough interfaces as compared to these elastic gels, where we see more or less a very clean interface in elastic gels. Bubble sizes and shapes, interestingly, didn't seem to change as much within the same class of elastic gel or viscoelastic gel, regardless of the inflation pressures. And more or less the size seemed to be similar, and the points at which these have been plotted are one just before the bubble popped inside the hydrogel. So Anshul has gone back and measured with Ibn the R effective. So by fitting that shape to a sphere or an ellipse. And here are the results from the viscoelastic gel in red, and the blue ones are those for the elliptical gel. And we see for one class of all the inflation pressures, these are done at various inflation pressures from 500 to 8,000. And in general, we see the same trend of how the radii increase as a function of time before the bubble pops in both gels. They're also very repeatable. And what you see below are similar curves for the aspect ratios, where we are showing that regardless of the size, in one case, they try and become more spherical in the viscoelastic case, whereas they go through this very sharp dip before they increase and pop. And that seems to be a trend of what we see in all our more stiffer gels. Either it is the MgO or in this case, the elastic gels, where we see very similar trends. The speed with which the crack runs is also shown here. And the reason why we are trying to do this is because we are thinking of modeling this. And so we have a shell here, and we are asking we are asking the question, how can you grow this either in terms of the area or by stiffening of the matrices due to the strain stiffening effects that could come about inside the hydrogel to cause slight differences in the stresses that can evolve inside this material. So before I talk about the model, I will tell you how the critical pressure, and these are, again, some of these are pilot results. We need to do a few more additional experiments before we can say conclusively what these are. But in general, we see a very nice trend that the critical pressure for cavitation is about 12 kPa for the elastic gels, whereas it's about 16 kPa, not super different. But the GC values are extremely low for these very soft gels. These are almost 2 to 3 joules per meter square as compared to an order of magnitude difference that we saw with gelatin hydrogels. And so there we had almost 35 or, sometimes we had 25 to 50 or so. That was the range that we got for our gelatin samples. So we are able to, and second most important point is that the critical pressures that we got are more or less the same inside all the hydrogels, regardless of the inflation pressure. So although we expected that the viscoelasticity would contribute significantly to these effects, it seems that regardless of the inflation pressures, we seem to get very nice results for the GCs and as a result of the PC calculation. So this brings us back to what is the role of surface tension. And so these are our thoughts, and I'm happy to hear you out if you have additional ideas of how this can be done. So what we are trying to do now is to create these prismatic rods, which have slightly different curvatures. So we want to use these prismatic rods of elastic and viscoelastic polychromide of slightly different cross-sectional areas, with an idea that if you are trying to, once we release these rods from the mold, which is a 3D printed block, we are essentially looking at a balance between the restoring elastic forces and the surface tension after it is removed from this mold. So if we will keep track of the height, which is more or less the same in these two cross sections, which are in the left case of purple, it is an equilateral triangle, whereas in the right, it is an isosceles triangle. The idea is that this blunting will happen where this prismatic rod is going to try and achieve a more circular shape. And so we will watch how the heights, the lengths, and theta, or the kappas in this region start changing as a function of time. Based on the theory of thin elastic rods, we can look at the balance between the stretching and bending in the prismatic rods, which arise from the resultant forces and moments with respect to the centroid. And clearly the centroid is going to be very different in the isosceles shape. And so we want to calculate the curvature, compare it with the linear theory with the experiments that we have, and estimate if they follow these kind of trends, which will tell us how dominant is the surface energy term in our expression for failures in soft matter. A second thing that we are looking at is how can we model bubble growth, which is looked upon as almost as inertial B-like stresses that we can estimate in this model. So here we have initial as the bubble grows from this one to outside, and this box shows that it's an infinitely large material, and so we are looking at how this blob is growing inside this material, which is soft rubbery. And so we know for all rubbery materials there is a limit point instability. So the question is, how will this limit point instability be different in both these control and NGO gels, which is something that we want to estimate with the model. We also look at growth of this region, which is air, and this outside gray region, which is all polymer. And we want to look at it as looking at the kinematics of growth as a deformation gradient, which is a multiplicative decomposition of the elastic term A and the G, which is the growth tensor. The growth tensor is something we can estimate based on all these changes that we've been measuring of how R changes as a function of time, and so on and so forth, where we can estimate a growth tensor as some G of R, which we can get from our experiments. So our idea is that we can look at this limit point instability and measure and then compute what the tensile residue stresses and the compressive contact stresses are in this very large infinite medium by calculating the Cauchy stresses. The thing we are missing in this expression is again the stresses inside the material. So what we've also done is we've gone and done some tried to get the strain fields ahead of the crack tip. So what you see here are experiments done with two synchronized high-speed cameras. We typically are using only 2000 FPS, which seems to work well for our elastic and viscoelastic gels. And here you see these are very nice and whitish with these black dots that they've been able to smear really nicely inside the 3D volume by first plasma treating these silica beads and then curing them to get this uniform beads. And here is that bead where you see that it's a little more milky as the bubble in the viscoelastic gels. So these are two cameras which are offset by about 40 degrees, and we have synchronize their acquisition. And we now, these were done in NCBS with help of a colleague and his student Girish, and who has been very helpful with getting some of these pilot data. So we want to try and now use DVC, digital volume correlation, to see if we can estimate what are the stresses, especially ahead of the bubble inflation, using these positions of the various beads that we have smeared inside the sample. And hopefully that is the expression that we use in this value of the Cauchy stresses that we have calculated theoretically. So that's where we are going with the study. So to conclude, we think cavitation rheology is a really nice, useful method to estimate toughness of soft materials, with some issues that it cannot be used to estimate GC in strain stiffening materials or when using Ogden, Mooney, Rivlin-like strain energy functions. If we are using just a simple Neo-Hookean model, it seems to work beautifully. Notch tip blunting seems to be a characteristic of soft matter failures, which came as a complete surprise to us. So we showed that cracks propagate in soft materials when the tip seems to be at its blunted position, which is also something new that we didn't know earlier. These MgO crosslinks are very useful to increase the toughness of gelatin hydrogels when we can't use this additional glutaraldehyde. So these are experiments we hope to set up very soon to incorporate cells inside these gels to estimate the 3D mechanobiology. Last, we are very excited about these results on the elastic and viscoelastic hydrogels, polyacrylamide gels, because they are allowing us to tease out the effects of both the surface tension and these elastic/viscous properties that seem to contribute in cavitation rheology. So where are we going with it? So Neha in the lab has been doing a lot of the stretcher experiments using our stretcher, and also a new student, Santosh, who has been able to cure these polyacrylamide gels in our mini stretcher, and we are now doing experiments to estimate how much traction stresses individual cells exert when they're being pulled either in a collective or a single cell. So we're really excited about some of these results that are coming out in mechanobiology. So that is our stretcher. And I want to stop and thank everyone. So these is the lab members. Specifically, Anshul is over here, who worked closely with first study was Anshul alone, then with Supreet. Even though she doesn't seem to be in the picture, which I just realized today, so we need to do justice and take a new picture in the lab. And he's an MTech student who will be finishing soon. I want to thank IISc for intramural grants, DBT and IISc DBT funds, which supported some of this grant, and also SERP. Finally, thanks to Sanjay and Shushmita, some of the early experiments on gelatin were done in her lab with high-speed geography. So I'll stop now, and I'm happy to take any questions.
Okay, thank you, Namrata, for a very, illustrative lecture that you have given on this new topic. I see a lot of questions and also some kind of discussion that's going on in this chat box. So shall we take them on in a sequential manner?
Sure, anything, however it works.
Yeah. So I think first there is one from Pabbu Krishnamurthy who is asking, GC is the critical energy for fracture.
Yeah.
Can you put some light on how to calculate it experimentally? Any standard test procedure?
So all the things that I have talked about were done experimentally. What I want to highlight is that there are various terms which are being used. What are critical is either this, I'm sorry, I'll just go back to the right slide. Let me see if the summary slide has all my information here. So it doesn't have those expressions. I'm sorry. So I'll have to just go back and find those. So in the case of cavitation rheology, GC is determined primarily by this value of PC, which is the critical pressure at which the crack fails. And like I told you that that we have to go closely hand in hand with experiments and analytical models. And here we are showing there are some problems with using these kind of linear models to estimate failures. So if we can get the expressions of PC, then we are able to compute the GC in this case. In the case of single edge notch and pure shear experiments, we have derived expressions that are that have been published in literature, and so we have used those to calculate the GC, which are shown here. So G is this d psi by dA, which is something that we use, and it was done routinely many years ago, 1963 by Greensmith. So these are standard tests, like what I summarized. So either it's single edge notch test or the pure shear notch test, where again we have slightly different relationships for how you write the GC, and that's how we can experimentally quantify the GC using these data. I hope I've answered your question.
I guess so. And there is one from Vipin Chandra who's asking, could you please clarify at which strain level the spatial results displayed in the slide number 21 were captured for SNAP?
Okay, could you please clarify at which strain level the spatial results were captured for SNAP? So this was done. So I think he's asking at which point during the propagation, right? So you can get this kind of maps at various points during the progression of the crack. At a single time snap that I have grabbed here, we have watched slightly above on this line, red line. We've also looked at the green. I didn't plot that, but there's another point at which we plotted this because that's more obvious. So we see a very similar pattern of these ABCD domains as the crack progresses inside the sample. When we come towards the end of the sample, it looks a little complex, and so we don't use this domain as much. So you see a very similar domain of ABCD throughout these time snaps that we see. Here specifically, I have used this first point when the crack grew. Immediately when the crack grew, so like I said, it stalls for a very long time at a certain length, and then it runs inside the sample. So just before the run is when we have calculated these parameters. which are plotted in slide 21. But that's a very good point.
Yeah, I mean, if I might just want to get clarified, this region D that what you have indicated, it
Yes.
also reflects the edge effect in some sense.
Right. That's right. So it is the edge domain, and that is why it looks very complicated.
Then would you say that there is some? Please go ahead.
Please continue. No, I'm sorry. I don't want to cut you off. Please complete your question.
Yeah. See, if that is the case, then is there a kind of a valid crack length over which these results are kind of to be considered? Like normally in fracture mechanics, we say A by W of something.
That's right. So the pure shear test, so that is a test that is why we try and look at the strain fields. So if I look at my videos, let me see the part where I show you. Yeah, this is the one I wanted to see. So if we are looking, so each of these points are where where typically the crack is run and it has stalled just before its next run. So that's why we have these points here. So as you have torn the sample almost 3/4 into the sample, you start seeing this region D is hard for us to delineate. So if we are looking, so these are not my configurations or my choice of geometries. These have come from classic fracture mechanics ideas on sheets of rubber. We've only adapted it for hydrogel-like materials. So at which point does it stop? As we are getting almost beyond the 50% mark, you will start seeing that this domain D starts looking more and more thin and short. And that is when we'll have to essentially not take anything further to model. But this A by W, the rules of how long should the crack be inside the sample, that is also something that is followed from literature. Does that make sense, Raghu?
Yeah, fine. Thank you. So I'll just continue. I think Dr. Datta has asked, can numerical simulation be done with FEM? If you have any knowledge, please share the reference.
So yeah, people have done numerical simulations. It's so most of the simulations are easier to run on. So we are hoping to start some of this work, and in fact, some of the cavity growth is something where we will need to do some finite element models where we are using an abacus model. I'm happy to share the references we have. I don't a priori have it right now in my head, so I'll have to go back and check that. But there are some papers which have looked at how bubbles can grow ahead of the crack tip. I know that a lot of this literature comes from, so there are at least 3 or 4 papers. I think Hui has done some very nice work. See Hui, and there are others who have done, and I'm happy to write that later if someone wants to reach out.
Yeah, I see Vikram has raised his hand, so.
Okay, sure.
I just wanted to get this straight. The crack here starts off sharp, it blunts, and then when it propagates, does it resharpen or does it propagate as a blunt crack?
Yeah. No. You're right. So it's a very good question. So I want to say it starts like a very sharp crack because we've used a razor blade to notch it. It's very sharp. And when it propagates, I'll show you the graph of kappa. I don't know where that is. Yeah, so the curvature is very high when it starts out. Okay, then as it goes through time, it drops to its minimum value. Again, you see the curvature increase, which means that as soon as it has run inside the sample, it's again sharp. Then again, it goes through its blunting at various time points. So it seems this.
Now, I was going to ask, is the blunting some form of rheological behavior and micro void formation ahead of the crack tip, which then coalesces into some sort of damage, and then the thing pops in and then repeats?
Right. So we looked, there is no microvoids which happen in these materials. We see those crazes, which are classic in polymers that we also see in gelatins. We think that it's the surface energy term which seems to dominate this response. of how that blunting behaves. And this is seen in most soft matter. I haven't discussed my work on cells, but even cells have this blunting behaviors before you create very sharp regions where they start migrating. So it seems like the surface energy dominates the response in soft materials. So there is no microvoids as far as I can see in our videos.
And there's no mass transport here. I mean, there's no fluid flowing from one region to
No.
another.
We haven't been able to. These are all completely hydrated. So these are two-phase materials. They are not drying out or anything because the experiment is done very quickly. I don't think there is a fluid transport. So in terms of the kinetics, I think it is more of the surface energy which dominates this response rather than any diffusion type behaviors.
Thank you.
But we don't know for sure. I think we'll have to finish our experiments to measure surface energy, which seems to be a little more tricky than when we set it out for. So we're now developing our ideas towards that. Yeah, but those are very good questions.
Okay, I think I'll continue with the other questions in the chat box. Amol Pupuluri has asked, would you like to know how GC is calculated? Also, why is the test called a pure shear? As I can see a mode 1
yeah.
test there only.
So that's why we put this figure, it wasn't very clear, I think, when you started with. So it's when we are tearing this film, we are not able to have eyes on the ZZ direction. But these are called pure shear because you create this pure shear region in this YZ direction. This is like a mode 3 in the YZ direction. And you can see it based on the strain calculations that we have also seen, that the strains, if you look at the EXY strains in both the gel types, you will see that you have a pure shear maintained almost consistently until the end of the test, where already I told you there are effects of boundary and others coming in. But here you see this very nice, the shear strains, and you don't have much else happening in the other components. So that is why it has been called in literature as a pure shear test. So again, so from this, how was the GC calculated? GC was calculated using an expression that we've again pulled from literature, and that is this one here. And this is also this is something that we have our work is in review, and hopefully we hear back soon. So those are, I think, my answers to Dr. Amol.
Okay, thank you.
I
And the
think it
next
was the
one
same.
is,
Yeah.
I think if it is mode 1, it is pure normal test with the DCB test.
Yeah, so I think again, those are similar questions on the pure shear.
Yeah.
And I think again, this slide should help clarify why it is called a pure shear, or also this picture that shows where the pure shear is valid. Otherwise, it's confusing because it looks like a mode 1, but actually the pure shear is introduced in the YZ direction. And what you're viewing from above is the XY plane, which is why this confusion arises, I think.
I think similar discussion. Is it easy to do a DCB test in soft materials to compute GC?
They are very soft, so sometimes they collapse under their own weight. It's not easy to set up these experiments. So I mean, it's worth trying, but I think I have a lot of, I feel that this pure shear test is actually really fantastic because we have such a nice, careful, controlled tear inside the material that you can visualize many of the processes at many different scales. If so, to address the kind of questions that Vikram asked, is there any effect of water? All of it can be done very nicely if we start, splitting that question into many parts and still using the pure shear test. So I don't see the need for doing any other additional tests. And also, I think, doing 3 different tests within one PhD thesis was quite a bit of work. And that serves as a very nice comparative fracture approach to testing failure behaviors in soft materials. I don't think that, so I don't find the need for doing it. Perhaps it could be done, but I haven't seen the beauty of doing it yet.
Yeah, there are a couple of, kind of exchange of, the information between Amol and Krishnamurthy. So I'm not going to go through that. Dr. Sitaramu has complimented you for the wonderful presentation. And Vipin Chandra
Thank you.
has asked, how is it possible to use the optical cameras for DVC?
So we are looking at it because it's a really small domain so ideally you're right that we want a confocal stack. But the challenge is that you have very thick gels and there's not much that you can do, and the cracks progress really fast. So to have quick confocal images are difficult. So we are looking at beads which were visualized in the vicinity of the crack. So we are able to extract some of the information, but I agree that for the entire range of deformation, we might not be able to track every single bead inside that blob. That's pending work. The field of view in both X and Y and Z, because these are really high-end cameras, so we are able to get over, many pixels that we are able to track. But I understand your concern, and we really need to do those experiments to finish the quantification to see if it can work out. In the small domain, I think we can get some estimate of the strain fields. We might not be able to get the entire domain until the bubble pops. But the reason why we are doing this is to get the initial zone where we can compute what the shear stresses are, what the Cauchy stresses are, even if we take a linear approximation. So even that part has not been done in the literature properly. So we are hoping to contribute to some of these ideas in literature. But it's an excellent point, and I see the challenges, and we haven't got an answer just yet.
Thanks. And there is a question from Dr. Sundar, who is asking, is blunting a sign of buildup in resistance to crack extension rather than a reason for crack extension? And he says in metals, it is the former.
I don't see his question. So we have thought of blunting purely from the perspective of surface tension related effects. We don't see the surface tension effects contribute too much to metal failures, but we are now thinking that maybe in soft materials you have this surface tension because you have this gel air interface which seems to drive or uh, create more and more of those rough edges in really, really soft materials. And that is something where we know that surface tension contributes. So we are looking at various length scales where this could operate, where these elastocapillary effects work. And so we are trying to design our prismatic rods to understand this better. So I still haven't got my answers clear. And so I think Sundar's question is excellent, but I'll have to get back to Sundar when we next meet to chat about some of these ideas. But thank you very much.
Yeah, then there's one from Deepa, who is asking, very nice work and lucid presentation, very relevant in bio inks in 3D printing. Your thoughts? I think she's wanting to know whether it's relevant for bio inks in 3D printing.
Yes, absolutely. So as we are trying to so it's not just for squeezing the gels and getting it to go. We are looking at cases where you've not just bioprinted a part, but you're exposing it to a certain mechanical load. And this is where most bioprinted materials fail because there is no residual stress and the cells have not been able to respond by creating a certain residual stress by itself. So just printing, in my opinion, is not going to give you the kind of tissue engineered parts that we really need as replacements for our poor livers and poor kidneys. I think we need to think more creatively of where gels, cells, and bioreactors are closely working with each other before they can create organs and tissues of interest. So the next few years are going to be dominated by how gels and cells will interact. So I think much of your question is very relevant, but I don't have a full answer for that.
Well, I think others are complimenting you for the excellent talk. And so, I mean, before I kind of conclude, I would like to know, see, you have measured the strain using the DIC.
Yeah.
And for both elastic and viscoelastic materials. What was the kind of strain rate that you had in the case of either elastic or viscoelastic materials?
So I think you mean with these viscoelastic. So we have done various strain rates. So our bubbles, so sorry, here. So the inflation rate, which sort of dictates the rate at which I load my gel, changed all the way from 500 to almost 8,000 microliter per minute. So this is a loading rate that we are putting on how the bubble is growing inside your hydrogel. So, and this was done for both these elastic and viscoelastic gels. We don't have any DIC results for these kind of experiments because of the challenges in visualization of these gels. We are trying to address it through DVC. We haven't got our answers just yet. So what I want to clarify is for these materials, we have not done those pure shear experiments. So I think we moved to look at what really attracted me to this question was this shapes that seem to they completely change from sphere to these elliptical penny-shaped bubbles. And that transition, symmetry breaking transition is of much interest and what drives these transitions. So that's why we decided to use the cavitation rheology and not the pure shear test. So I don't have an answer for your question.
I'm probably asking a very stupid question. Do you think that the outcome of your work can be kind of considered for the case of self-healing polymers to repair the composites or something like that?
Yeah, that's not a stupid question at all. So so first of all, we don't understand failures in soft gels at all. So, so there are very few groups which work on soft gel failures. And so the idea is if we can understand this better, then we can build this in with the kind of self-healing that you're talking about at a later point, uh, because you really need to break that capsule which contains this polymer glue that then cures itself under a specific load. So those are all additional questions, and it can also be used in those applications. For me, this was an essential question because much of my work has been on soft matter, and where we see these tears in vessels, and we don't completely understand. In this case, it is easy because it's isotropic. We haven't even introduced any anisotropic fiber layouts, which is where most of the questions are in dissections that happen in arteries and cause those kind of really horrible deaths in people. So those are questions that we want to have a handle on. But you're right, it can also be applied to self-healing polymers.
I can see a raised hand from Daksitaram.
Yeah,
is he
please.
alone? But I could see that he has already said thank you for the lecture. So probably he raised his hand and did not want to speak, so or could not speak. Okay, so if there are no more questions from the audience, then I think once again I see Daksitaramu raising his hand. Daksitaramu, you can go ahead. Daksitaramu? Looks like he's not able to speak. So anyway, so we will kind of thank Professor Namrata for this excellent lecture, which also attracted a good number of questions from the audience as well as from the organizers like us trying to get educated on some of these aspects. So thank you very much, Namrata, for this excellent lecture. So we will clap and give a
Thank you,
appreciation
Raghu,
for
Vikram.
her excellent lecture. Yeah.
Thank you, Namrata. Raghu, you want to announce next month's speaker if we have it?
Yes. I think next month on, we just look at it, May 20th, I think the lecture will be by Dr. Sundar, who will try to kind of compare between the aircraft structural integrity program and then the engine structural integrity program. So I think we all look forward to, your participation for the next month's
Yes,
webinar.
I'll be looking forward to that as well. So thank you very much. I really enjoyed, and a lot of my material was new. So I thank you very much for your patience and a very nice audience. Thank you, Raghu. Thank you, Vikram.
Thank you.
Bye.
Yeah, thank you.
Thank you. Bye-bye.
Bye.
Bye-bye.